Now in this section we will be studying a little bit about the brief history of microbiology. The ancestor of bacteria were present as the first life on Earth, and so these ancestrals were at least, the Earth is 4.5 billion years ago. The first ancestrals were discovered about 2.5 billion years ago, and we didn't know much about it till the advent of a microscope, which was developed by Robert Hooke. But he observed tiny little boxes on bark, and those he called as cells. Later, Rudolf Virchow studied cells and proposed the theory of cell theory. A cell theory states that all living things are composed of cells, and they all come from pre-existing cells. This actually refuted the old concept, which was of spontaneous generation. Earlier, people used to believe that life just existed by itself. There was a vital force that caused them, and things just grew without understanding what they were. When Van Leeuwenhoek studied under the microscope in 1670, he was the first one who actually saw paramecium and little tiny organisms floating in his feces and even in water and contaminated water. Louis Pasteur by 1861 then showed that microorganisms are in the air, and he refuted the spontaneous generation concept. And the way he did that was he took two flasks, and you can see up here there's one flask that is open up here, and the second flask has this curve. He placed a nutrient broth in both the flask, and then he heated them, and they were not sealed. Now, after some time, he found that there was microbial growth in the first flask and no microbial growth in the second flask. And this, he explained that these microbes that were present in the air fell into these openings, but they were trapped in this swan-shaped. So here are all the microbes trapped in here. So they don't go into the other column, and they don't contaminate the broth in the curved flask. So he refuted the spontaneous generation concept. Imagine how much opposition he must have had at that time. So when was the golden age of microbiology? The golden age of microbiology is between 1857 to 1974. This is the time where many different techniques were developed, and these techniques are actually used even now. For example, pasteurization was developed, and after it was investigated by fermentation. Pasteurization is actually heating, providing high heat for a very short time, and we are still using that for pasteurizing milk. Certain aseptic techniques were developed at that time, and these techniques were responsible, microbes were responsible for fermentation. So if aseptic techniques were used, and we will talk about aseptic techniques in labs, in which we're talking about how not to touch with hands, and how we use the loops to heat them. So those are using the gloves and so forth. Those are aseptic techniques. Washing hands is also an aseptic technique. Fermentation deals with conversion of sugars to alcohol to make beer and wine, and this was discovered by Pasteur in 1857. Pasteur later on had disapproved spontaneous generation. A couple of years later, he formed pasteurization concept. Then Lister was the one that proposed the aseptic technique. Later, Lister was followed by germ theory of disease, which we will look in the next slide, and Koch's pure culture. So let's look at the germ theory of disease. Germ theory of disease was proposed by Robert Koch in 1877. He actually did experiments in which he showed that certain diseases were caused due to certain microbes. He had heard about this girl who was working in farms and did not get, she was working with Cowpacks. She was exposed to Cowpacks, and she did not get Actually, that was Edward Jenner who had done the immunology, so I'm mixing it up. Koch had actually done experiments in which he proved that Bacillus anthrax caused anthrax, and it was later Edward Jenner who had studied this girl who was exposed to small cowpox, and she did not get sick, and it was because she had immunity towards smallpox, because smallpox was a disease at that time which was very widely being spread out in the world at that time, and many people were dying because of smallpox. So what about modern microbiology? Alexander Fleming is the inventor of the modern microbiology, because he discovered penicillin by an accident, which was made. Penicillin is made by a fungus, and it's a chemical that is produced by one organism and it kills another one. For example, Staphylococcus aureus is a bacteria that penicillin can attack. Penicillin once was discovered, was tested very, very massively, and especially during the World Wars, it was very widely used. Some more terms up here. Bacteriology is the study of bacteria. Mycology is the study of fungi. Parasitology is the study of protozoans and parasitic worms. Just a reminder, you need to know all these definitions. These will all be in the test. Immunology is the study of immunity. Vaccines and interferons are being investigated that will prevent and help cure many viral disease. And lastly, virology, which is the study of viruses. Study of microbes also helps us in the process of bioremediation. Bioremediation means that you degrade organic matter in conditions where there's contaminant or in sewage and so forth. Especially we hear of those after a big oil spill or some drastic effect. We will hear about how bioremediation is used to degrade and detoxify the pollutants such as oil and mercury. Biotechnology is the study of DNA and how to manipulate DNA to and there are various procedures that are done in which we then introduce new genes into that particular cell. And we will do section on biotechnology and genetic engineering later on too. Lastly, what is normal microbiota? Normal microbiota means that those bacteria that is present normally on our cells they may be present inside or outside the body, and they actually prevent growth of many pathogens because of the pH changes and certain chemicals that they produce. Certain growth factors such as folic acid and vitamin K that they can produce. And when we do not have resistance to the resistance is the ability of the body to fight these diseases. So when you get infected and you don't have the normal good microbiota, then you have lower resistance. Resistance factors can be present on your skin, stomach acid, which will affect your pH, and many other chemicals that are like enzymes and coenzymes will be formed by the normal microbiota. Remember, I said 5% microbes cause diseases. A lot of them are good. And lastly, what are infectious diseases? Infectious diseases are diseases that take over a host resistance. So it's a bad pathogen that has it has destroyed the host's resistance, and the host cells will get destroyed, and the animal or the human person will get sicker and sicker. And they can emergent infectious diseases, or EID for short, are those that are widespread. For example, anthrax and AIDS and hantaviruses. These are all examples of infectious diseases. So this concludes our section on microbial world and you.